In our laboratories, we do a combination of electrophysiology, calcium imaging and functional measurements, all in isolated blood vessels. So we're particularly interested in how hyperpolarization of smooth muscle cells occurs, whether it be directly on smooth muscle cells or via the endothelium, and how when a blood vessel hyperpolarizes, how it dilates, and also how that dilation can propagate through the coupled cells to cause conductive vasodilatation. I love watching the microcirculation. The red blood cells flowing through the small arterioles and returning through the venous circulation is just mesmerising. And I really want to understand how it works and what regulates moment to moment where the blood flow goes within a particular tissue. We're particularly interested in skeletal muscle, so we're isolating arterioles from a skeletal muscle bed and trying to think how factors that are released during exercise can cause hyperpolarization and cause conductive responses. And for example, we're looking at ATP, which when it's added to the outside of a blood vessel generally causes constriction. But if it's released into the lumen of a blood vessel, it stimulates the endothelial cells and hyperpolarizes and dilates vessels. And we've shown that that dilation can cause a conductive response. So that's a really neat way of when tissue PO2 is low, that if red blood cells or other cells can release ATP, we can get a good functional hyperemia because of the increase in diameter of the blood vessels. One way of delivering ATP and potassium ion into the lumen of a blood vessel was actually first published in the Journal of Physiology. So that's what we call the triple cannulated arterial system, where we isolate out an arteriole, but in this case we include a side branch as well. And into that side branch we can introduce these agonists into the lumen of the vessel, and so in the case of ATP we'll stimulate the endothelial cells and stimulate dilation, which we've shown in the Journal of Physiology will cause a conductive response as well. I've got a fellowship from the British Heart Foundation, and the aim of that project is to translate our findings to humans. So we're collecting tissue from right atrial appendages and dissecting out the small arterioles and looking to see whether conductive dilation occurs in those human tissues, and indeed it does. And so we're very excited by that, and indeed this is a whole new approach that I think is going to have a lot of implications, because we can get tissue from diabetic patients, hypertensive patients, and try to establish what pathways occur and how they can be dysregulated in those disease states. I think the big challenge is to translate our findings more on a functional level to more molecular, more micro level. So at the moment we're developing techniques to study these small arterioles, to isolate them and in fact isolate the endothelial cells out of them and see which proteins are expressed using proteomics, and also using PCR techniques to precisely identify which proteins are potentially there.